This is cell membrane and metabolism. All right, we begin with the most important molecule for the cell membrane, the phospholipid. The phospholipid has a watery or polar or hydrophilic, water-loving head, and a fatty or oily or hydrophobic or non-polar tail. These are actually fatty acids that make this tail. And the tail is significantly longer than the head, so this is kind of a disproportionate drawing. So it's schizophrenic. I'm water-loving. I'm water-hating. I'm polar. I'm non-polar, and so on. And the fact that it has this schizophrenic character is all important, because look what it does. It lines up into a bilayer. The water-loving parts face outside of the cell. This is the extracellular fluid. And the water-loving parts also face inside of the cell, because the interior of the cell is watery fluid. And the oily, fatty parts face each other, the interior part of the cell membrane, and they are attracted to each other. They have a sort of a magnetic attraction. They want to stay together, and they're repelled by the outside and by the inside. And so they're quite stable. They won't change their orientation. They won't be easily broken. So it's a very ingeniously simple, yet stable and flexible. This moves around like the surface between oil and vinegar. It can, if you move it gently, the oil and vinegar surface won't be disturbed. Well, this is much, much stronger because it's a bilayer, a double layer. Here's a video which does a good job of kind of explaining that. So here's our bilayer again. Watery heads, oily tails, and there's this new molecule. I don't know if you recognize it from when we studied biomolecules. This molecule is a steroid called cholesterol. And cholesterol, you might recognize it, is a fatty substance. So what does it do? It increases the attraction of all this hydrophobic fatty, so to speak, all these hydrophobic attractions and connections. And so it strengthens this hydrophobic aspect of the cell membrane, and as a result, it prevents things from just entering. So most things that are floating around out here and down here are watery. They're water-based, like everything in our body, like most everything in our body, and they can't easily penetrate into the fatty part of the cell membrane. So there's a nice summary. Strengthens the bilayer and therefore decreases its permeability. Permeability is the ability for something to penetrate through. Diffusion is the random motion of particles leading to even distribution, and for the most part leading to disorder in a certain sense, because it's always going from high concentration to low concentration. It doesn't let things pile up, so to speak. A given molecule is going to naturally always go from high to low concentration. Everything goes from high to low. High to low is as natural and spontaneous as things rolling downhill. It's also called going with the concentration gradient. Instead, to go against the concentration gradient is as difficult as things going uphill. And things can go uphill, but it takes effort and energy. So the same thing is true with the diffusion of molecules in our body and across the cell membrane. So when things go downhill, it's called passive transport, requires no energy. When things go uphill, that is from low to high concentration, then it's called active transport because it requires energy. Okay, here is our bilayer again, and notice there's these massive, huge molecules represented by these pink things wedged into it all over the place and spanning it from top to bottom on both crossing both sides, and they are proteins. In general, they're just called membrane proteins, and there's a great variety of membrane proteins. We're going to look at 4 of them. These 2 proteins are both proteins involved in transport and getting things transported across the cell membrane. The first one is really easy. It has like a tunnel appearance, right? It kind of look like a tunnel, and it's called a channel protein because it's a channel for things to pass through. This is the way that water, for the most part, travels into the cell, across a channel protein made for water. This is another transport protein called a carrier protein. This one's a little bit harder to understand. This is supposed to be the same protein at 3 different moments. In moment 1, it's open to the outside and something can get in. In moment 2, it's shifting its position And here it says last position. It's kind of rocked back and forth until it got this new position, which allows this substance to enter the cell. Surprisingly enough, this doesn't need to take energy. This rocking back and forth can happen spontaneously without the use of energy. It actually can be both passive or active transport, depending on whether going from high to low or low to high concentration. I hope you recognize these same two. Here's the channel protein. Here is the carrier protein. And here there's high concentration, this blue solute, whatever it is, low concentration. So this is passive, so this requires no energy. Instead, this is a carrier protein again, but if you look carefully at the squares and circles, it's going against the concentration gradient. It's going uphill, and that's why it requires ATP. So this whole diagram is actually very useful and important. This is passive transport, and all of these are diffusion. As passive transport, meaning requiring no energy, they're all diffusion. These are substances which diffuse directly through the cell membrane, which are allowed to enter in. There's not many of them. They consist of oxygen, CO2, and lipids. For the most part, those are the only things that can do it. O2, oxygen, CO2, and lipids. And they have to go with the concentration gradient. Here are things that travel through a channel protein, but with the concentration gradient. Here are things that travel with the carrier protein. And so they're still diffusion, because they're natural and spontaneous, but they're facilitated because they're being helped or facilitated by a protein. And instead, when you go against the concentration gradient, it's active transport. Okay. Here is a view of another type of protein called a glycoprotein. And if you see, this little chain is a chain of sugars, and therefore it's called a carbohydrate chain. So glycoprotein has a little chain sticking out. And so here's a picture with many different glycoproteins, right? Here's a glycoprotein here, here's another one here, and so on. And what is the purpose of these glycoproteins? They have a double role, double function. They act as an ID. So these glycoproteins are labeling the cell, saying, I am a cell belonging to so-and-so, whoever you are. Or I am a cell belonging to this organ. So they're kind of identifying the cell, allowing others to recognize what it is. The other purpose of glycoproteins is this is a picture of two cells lined up. You can see their phospholipid bilayer here. And this picture is way more complex than, you don't need to really worry about it, but you just should get the idea that there's glycoproteins and carb chains coming out here. And look what they're doing, they're attaching. Because glycoproteins are involved in cell-to-cell adhesion, meaning in the attachment of one cell to its neighbor. For what purpose? Because cells are not just free-floating. Some of them are, like the ones in your blood are, but many cells are not free-floating. They're attached to each other to build tissue. Okay, our last fourth type of protein we're going to study is a receptor protein. Here's a receptor protein, and it has the job of receiving a message from a chemical messenger called a hormone. This is the hormone arriving. They fit into place perfectly chemically, which is why they bind. Here it is bound to the hormone, and the hormone has a message to give. And this lit up part is obviously just a symbolic way of saying it's transmitting the message. Because of that message the hormone is given, the receptor protein is transmitting that message and activating the cell. Another image here, the receptor protein is like a pink wrench. Here, the yellow thing, I know they've called it ligand, but we're going to call it hormone. And here's the idea that there's been an activation inside the cell. The secondary messengers traveling to the nucleus to deliver the message, so on. You don't need to worry about this mechanism, but you know that when they bind, the message will be transmitted to the cell. The fancy name for a message being transmitted to the cell or being carried across the receptor protein is signal transduction. Think of it as the signal or the message going across the cell membrane. And what might that message be? It can be lots of things. It can be, you need to start making this protein, you might tell the cell. Or you need to change your permeability and change some of your membrane proteins, close them off. All kinds of things that might be in the message. Here's a really nice summary, so you should pause and make sure you understand all of this and write it out. And here's a quick review. Receptor protein, the hormone would fit right in there. Channel protein, where it travels through. We didn't talk about this, but channel proteins can have a gate. so that permeability can be changed. So you don't need to know that, but it's kind of cool. There's the carrier protein. So the two transport proteins are the channel protein, the carrier protein. This one will rock back and forth. And here, because it's got a big carb chain attached to it, this is a glycoprotein. Notice the little cholesterols in there. And notice, of course, the phospholipid bilayer. Okay. This is a close-up of a carrier protein when it's acting as, when it's doing active transport, when it goes against the concentration gradient and it uses energy. It's often called a pump when it's doing that, because a pump is something which requires energy to move things, to transport things. The sodium-potassium pump is a really famous one that we'll study a lot when we do the nervous system. Your nerves would not work if sodium and potassium weren't continuously being pumped out in certain ways. Okay, this is worth reading. The most important is diffusion. Notice that's what oxygen and CO2 and lipids do. The facilitated diffusion with the membrane proteins, and osmosis, with this, which is the diffusion of water. Osmosis, the diffusion of water, is a little bit tricky, but this example here explains that when there is unequal concentrations, usually this red solute would want to move across, right? The red thing would want to move to the right to equal out the concentrations. If this membrane were only partially permeable and didn't allow for the red solute to move, what would occur? The water would diffuse, that's what osmosis is, diffusion of water, uphill against the force of gravity to equal out the concentration. Concentration is super, super important in the body. And here's what would happen to a cell. Here's a cell in normal concentration. This is a red blood cell, which has a kind of a donut-like indentation in the middle. So this is a normal, happy, healthy cell. There's water entering and water exiting. Here's a blood cell in very unconcentrated, almost pure water. And what happens to it? Lots of water enters it, and it will eventually burst. The fancy name for that is lysed. And here's instead a blood cell in a highly concentrated solution. The solute is very, very concentrated, and it will shrivel. You might think of your fingers in very salty water, which wrinkle up. Okay. Semi-permeable membrane leads to this surprising distribution of water due to the force of osmosis. This is called osmotic pressure. The higher concentration exerts osmotic pressure. It's like a force almost. Okay, that's pretty complicated. Here is a review. Actually, we haven't covered the last two. Let's do that first. Okay, things out of order. Okay, this is the last thing we need to cover. Exocytosis and endocytosis. So here is a vesicle full of a substance the cell has made and wants to secrete. Notice the walls of the vesicle are made of phospholipid bilayer, just like the cell membrane. This vesicle will move towards the cell membrane until it fuses with it. When it fuses, it will secrete the substance it needed to secrete. That's what exocytosis is, right? The vesicle fuses with the cell membrane, letting out its contents. Endocytosis is basically the opposite. Don't worry about this yet. Listen for a moment what's happening. Here, a substance needs to be taken in, so the cell membrane begins to pinch in, and then it pinches in all around it and creates a vesicle, and it has taken in the substance that it wants. An important distinction. If this substance is a solid, it's called phagocytosis. If the substance is a liquid, it's called pinocytosis. This is just kind of annoying vocabulary, except that it ends up being important because these words are used in anatomy and physiology. Notice again, vesicle walls must be made of what? Phospholipid bilayer. Okay, phagocytosis, it's ingesting a solid. Here it's a liquid, so it's pinocytosis. And here's a very important thing. Actually, I think this picture is better. This is receptor-mediated endocytosis. These receptors are capturing the solute, and it triggers the solute. It triggers the cell to do endocytosis and take in that solute. The most common examples of exocytosis are secretion. Here something's been made in the Golgi bodies, it's being secreted. So exocytosis is involved in the secretion of enzymes, of mucus, of saliva, of all kinds of things. And the most common example of endocytosis involves a white blood cell ingesting a virus or a bacteria in order to digest it and kill it. So those are really common and important examples of endocytosis and exocytosis. And I'm going to go back and on the page, which is the summary. Here's the summary that you can look over and make sure you can name them all.